What's up, baby bio penguins? So, hi, coming to you because it is Friday, FRQ Friday. Yes, I'm a little earlier than I'm supposed to be because I just found out I have a meeting. So, let's go ahead and get into this because why not, right? And just verify that you can actually hear me because I've had earbud issues lately. So if you can't hear me, just make sure you let me know, okay? So, hi, we're going to do these FRQs, which I apparently forgot to make myself nice and big. Sorry. Okay, nice and big now. So we're going to do 2016 number 7, 2015 number 4, and 2019 number 3. I thought that this was last week, and then I messed up and I did the wrong questions. I don't know how I messed up, so I'm sorry that I did. So this is actually last week's one, but this is this week's one, so you get the idea. So we're going to do a lot with mitosis and meiosis with this week. So a certain plant has a diploid number of chromosomes of 4. 2n equals 4. Flower color is controlled by a single gene. The green allele is a capital G. It's dominant to the purple allele, which is a lowercase g. Plant height is controlled by a different gene, which the dwarf allele, FYI, that says dwarf, not short. That's going to come into play in a second. Dwarf allele is a capital D, is dominant to the tall allele, which is a lowercase d. Individuals of the parental generation with the genotypes capital G, capital G, capital D, capital D, and lowercase g, lowercase g, lowercase d, lowercase d. So basically, a homozygous dominant and a homozygous recessive are crossed to produce the F1 progeny. And they've given you this diagram. And FYI, just so we're looking here, okay, we see already our sister chromatids. So we've already gone through interphase and we've made the copy of our DNA. So we're looking at our homologous chromosomes, right? So here would be the paternal, here would be the maternal, and then the same thing here where we have paternal and maternal. So that's chromosome 1 and that's chromosome 2. Notice that they're different sizes. Chromosome 1 is long, chromosome 2 is short, okay? That's going to come in play in a little bit, I promise. So the first question we have is construct a diagram below to depict the 4 possible normal products of meiosis that would be produced by an F1 progeny. Show the chromosomes and the alleles they carry. Assume the genes are located in different chromosomes and the gene for flower color is on chromosome 1. Okay, so what they're telling us to do, okay, is to make sure that we can draw out what the 4 different gametes would look like from the F1. Okay, and this causes a lot of trouble for students, okay? Students on this exam, which actually this was one of the questions I graded, they drew all kinds of different things. Number 1, you do not need to be an artist, okay? You just had to make 2 lines, one long, one short. The long one was your chromosome 1, the short one was your chromosome 2, okay? And then you had to be able to appropriately identify which ones were the dominant alleles and the recessive alleles, and then of either the G or the D. Okay, so there doesn't need to be a huge artistic component to this. You just had to put some stuff down and make sure that we as the readers could tell what you were drawing. Okay, and again, these are our different gametes. Oh, I didn't have the answer. So we were trying to find the F1 progeny. So if our parents are homozygous dominant and homozygous recessive, then we know that the F1 is going to be heterozygote, right? Because the only thing that this parent could pass on would be that capital D. The only thing that this parent could pass on would be the lowercase d. The only thing this parent could pass on would be the capital D. The only thing that this one could pass on would be the lowercase d. So we were only making basically a dihybrid. Okay, so when we draw this out, we know that in our gametes, we're going to end up with 1 of each chromosome, right? I'm going to have 1 long and 1 short. So just go ahead and draw it out where you have 1 chromosome 1 and 2 in each of your cells, okay? So now, because of the fact that we know that it's heterozygous, we know that 2 of them are going to be capital G and 2 of them are going to be lowercase g. Okay, so this was the easy part, right? You draw out the chromosomes and you identify 2 of them as capital G and 2 of them are lowercase g. The problem now comes into play is now we have to come up with every different combination we could make, right? Because of independent assortment, you could end up with the 2 dominants together, or you could end up with the 2 recessives together, right? Because of independent assortment. But you could also get them crossed up, right? Where you could have the dominant of 1 and the recessive of 2, and the recessive of 1 and the dominant of 2. So you should have had in your cross, you should have ended up with basically 2 dominant alleles, a dominant and a recessive and a dominant, and then the 2 recessive alleles. And so all they looked for was that each one had chromosome 1 and a 2, that it was not duplicated. So notice that we only have a single chromatid here, okay? And that we had to have the combinations of GD, of all the different GDs, okay? Now, students on this question, make sure that you actually drew your chromosome. It tells you to draw it. So if all you did was give me the letters, we couldn't count that because you didn't draw any chromosomes. If you incorrectly drew the chromosome in which you still made it duplicated, you couldn't get credit for that because you weren't demonstrating that you understood that the sister chromosomes come apart. So this is one of those graphical kind of diagram questions that you might see as question number 5 on your exam. So remember, they're going to have you draw something if you're doing the paper exam. If you're doing the digital exam this year, then you will not have to draw anything. So they're going to somehow embed this some other way. They haven't actually told us how. So the next part of this question, part B, we had to predict the possible phenotypes and their ratios in an offspring of a test cross with the F1 individual and a homozygous recessive of both alleles, so double recessive. Okay, so students don't understand what a test cross is. They told us to, right? We're saying there's a test cross between the F1 and the homozygous recessive, right? So we know that our cross is going to be between our heterozygous individual from the F1 and the homozygous recessive, right? So if we do that cross, here's our heterozygote for G, homozygous recessive, giving us 1/2 is G, 1/2 is purple. And then here we do our heterozygous for dwarf, homozygous recessive, giving us 1/2 dwarf, 1/2 tall, okay? And remember, if you have questions, you can pop them down into that little question box. And so we're not done, right? We haven't figured out a ratio yet because I know the ratio of green and purple and dwarf and tall, but I haven't actually figured out the ratio of green dwarf, green tall, purple dwarf, and purple tall. So you have to multiply in order to get that, right? So you're going to do the 1/2 from the green times the 1/2 of the dwarf, giving you 1/4. And you do that exact same math for every single one of them, doing 1/2 times 1/2 because we have a half that are green, a half that are tall, a half that are purple, a half that are dwarf, a half that are purple, and a half that are tall, okay? And you were supposed to give us the ratio, okay? Now, here's one of those components where you need to make sure you write complete sentences, right? You naturally want to give me that ratio, right? You see it. The ratio is a 1-to-1-to-1-to-1 ratio. I said 4 ones, right? Okay. And so what students tend to do is just write out that ratio. Well, number one, they said phenotypic ratio. So you have to make sure you tell me that we have 1 and then write out your phenotype. Now, the prompt said dwarf. So if you wrote short, you don't get credit for that because you didn't read the prompt. Okay, so make sure you're reading your prompts. And if they give you a specific term, give them that term back. Okay, it's a green dwarf, not a green short. Green dwarf. Yes, we know that a dwarf plant is going to be lacking the GA3H in order to grow, right? Which we looked at a couple weeks ago. But that has nothing to do with short. We are saying it's dwarf. We're using our corrective terms from the prompt, okay? So make sure you write it out as a one, you would write out the ratio is, and then you write out the thing, okay? Now you could do this as a fourth to a fourth to a fourth to a fourth to a fourth. You could do 4, 4, 4, 4. It didn't really matter what ratio you chose to do, as long as you tell me that they are all equal, okay? So that is our first question. We're moving into our next one. Hey, look, it's Rhea. The part C is taking into account our linked traits, right? So if the two genes are genetically linked, so they're linked together, they're on the same chromosome together, describe how the proportions of phenotypes of the resulting offspring would have differed than those from the test cross. So we found our test cross that we would have had basically equals of all the different options, right? But because of the fact that they're linked together, it means they're on the same chromosome. So if you get the chromosome with the green allele, you're going to also get the chromosome with the dwarf allele. If you get the chromosome with the purple allele, you're also going to get the tall allele, okay? They were genetically linked together. So you would have found more than 50% would have looked like the parental units, right? And less than 25% would have looked like either of the homozygous recessives, okay? Less than 25% would have been the recombinants, right? Because recombination has to do with crossing over, and so you would have found that crossover could have put the green with the tall and the purple with the dwarf, okay? And so that's exactly what the scoring guidelines had. You could have said majority or greater than 50% of the parental plant phenotypes. You could have also said that less than 25% would have been green tall and less than 25% would have been the purple dwarf, okay? So I hope that was helpful. that's our first one. We've got 2 more coming. And remember, if you have questions, you can pop them down to the little question box. I know this is a unique time, so if you come up with questions after the fact, you're more than welcome to comment on the video, and I'll answer your questions later. So question 2015, number 4 is what we're going on to now. So both mitosis and meiosis are forms of cell division that produce daughter cells containing genetic information from plant cell. So we're going to describe 2 events that are common in both mitosis and meiosis that ensure the resulting daughter cells to inherit the appropriate number of chromosomes. Okay, so how can I make sure that in mitosis and meiosis I get the correct number of chromosomes? And so first thing you have to think about is that mitosis and meiosis occur after interphase and before cytokinesis. So everything you're currently thinking about that happens in interphase or cytokinesis cannot be given here and saying that it's part of mitosis. You could say that they both go through interphase before, and in interphase you duplicate your genetic material. You could say that they both go through cytokinesis after, and that's going to equally split the cytosol. Okay, but that itself is not part of mitosis and meiosis. Okay, so you got to think through the different parts and how those parts are going to allow you to get appropriate number of chromosomes. So number 1, think about prophase. Prophase is when we're going to condense our genetic material, right? Our sister chromatids are going to pair up. Our homologous pairs will come together in the meiosis 1 and prophase 1. And so the condensing of that chromatin is something that happens in both of them, and that would help to ensure we have the right number of chromosomes. Lining up on the metaphase plate that happens during metaphase, right, in the middle of that cell, that ensures that we get equal numbers. Ensuring that the kinetochores are attached to each of our chromatids ensures we get appropriate number. In anaphase, when we talk about that the sister chromatids or the homologous pairs move apart, that's also going to ensure we get the correct number. Okay, so any of those were definitely possible options. Other additional things you could have said that I didn't remember just now. You could have also talked about the M checkpoint. So remember that checkpoint involves that every single chromatid is attached to kinetical microtubule to ensure that we get equal division. Okay, talking about that you have the microtubules attaching to your chromosomes. Here is where we said prophase, how the chromatin condenses. Here we're talking about metaphase, where they're lining up on the metaphase plate. Here's anaphase, where they're coming apart. And then, of course, you could have said replication occurring before mitosis and cytokinesis happening after mitosis. Okay, so if you had just said interphase and cytokinesis, you would have not gotten credit for this because you didn't demonstrate that you understood that happened before or after the cell, like the mitosis or meiosis. Okay, so the second part of this question is where we're going to look at the genetic composition that's produced by mitosis differs from that's produced by meiosis. And we are describing 2 different features that lead to differences, okay? So it's overall asking, what is different between mitosis and meiosis? Okay, and most of those differences really come down to the fact that meiosis go through 2 divisions, right? So let's first think, how many divisions do they each go through? Mitosis goes through 1 division, meiosis goes through 2 divisions, right? You only replicate your genetic material once, but then you divide twice, which leads to haploid cells in meiosis. Since you duplicate once and you only divide once, that leads us into diploid cells in mitosis. So one difference is that we have diploid in mitosis and haploid in meiosis. Now notice, I said both of them. Whenever they ask you to contrast something or compare something, you need to mention both things. In mitosis, this happens. In meiosis, this happens. You must mention both, okay? So you could also talk about the number of cells, right? In mitosis, we make 2 cells because we only go through 1 round division. In meiosis, we make 4 cells because you go through 2 rounds of division. And then think about the specific things that happen, right? In prophase 1 of meiosis, crossing over occurs. In mitosis, crossing over does not occur. Yeah, you even have to do it there where you say that something does not occur in the other one, okay? Yes, this live will definitely be saved, and it's in the IGTV as well as it will be saved to YouTube. So then we also have, I already said crossing over. We're independent assortment, right? Independent assortment is going to occur in metaphase 1 of meiosis, but does not occur during mitosis. So again, we have to give those differences. So now, something I want to warn you about is that oftentimes in these questions, you're going to see where they've said like 2, right? They want 2 things, right? So as a student, you just start writing things, right? You're like writing some information. Well, you want the reader to know what your two are. Because if you accidentally say something in your first sentence that's wrong, right? But it's supposed to be part of your first thought, they're going to count that as 1 and then 2, and they won't read, they read past it, but they won't count past the first 2 you give. So be very clear. Say, the first difference is, and then write out whatever you feel the difference is. The second difference is, and then write out whatever that difference is. Okay. And that ensures that we as the readers are looking at what are you calling your two differences. Okay. Because in this case, you definitely could have fallen into a double point accidentally, right? There are 2 rounds of division in meiosis, which leads us into 4 haploid cells. In mitosis, there is 1 round of division leading us into 2 diploid cells. That right there would have been 2 points because you've both demonstrated that you know the number of divisions, the number of cells, and you've also mentioned haploid versus diploid, okay? So that is essential, okay? Oh, and something I didn't mention, chromatids are separating during mitosis and they do not occur during meiosis 1. That was another difference. Sorry, I didn't mention that one before. You don't have the whole rubrics memorized sometimes. It's okay. You're also not expected as a student to know every single answer on these things. They put multiple options so that you can kind of hopefully get one of those or 2 of those. Yay. So I hope that was helpful. And moving on to our third question. I have no clue how much time I have, but I know my meeting's starting soon. So 2019, number 3. Let me let you know that we are jumping back a little bit and we're going back into cell respiration for a moment. Right. So when I built the huge 245 page review, I asked students, do you want me to organize it based on where most of the content is? Or do you want me to organize it based on where the last content that you would need is? And they said based on the last content. So this question has a genetics component to it, but the meat of the question is a cell respiration question. So we're going to jump back to cell respiration for a second and just kind of bring all that back to the surface and connect it, because your questions are not just going to be from one unit. They're oftentimes going to be 2 or 3 units that have all been, connected together. So 2019, number 3, pyruvate dehydrogenase, PDC, catalyzes the conversion of pyruvate to Acetyl Coa, a substance that's found in the Krebs cycle. Ooh, and they tell you both words. That's nice of them. The rate of pyruvate conversion is greatly reduced in individuals with PDC deficiency, a rare disorder. Identify the cell location where PDC is most active. Well, first, the question tells me PDC works in this process here of pyruvate to Acetyl Coa, right? And that happens during the Krebs cycle. Remember, I told you it's important to know where things happen, right? So this is one of those, where does it happen? Where does Krebs cycle happen? Well, Krebs cycle takes place in the mitochondrial matrix, right? But they were nice to you on this question, and you could have just said it happens in the mitochondria because you understand that the Krebs cycle takes place in the mitochondria, okay? Sadly, if you said mitochondrial cristae, you do not get credit for this because you were too specific and gave me wrong information. So I don't really have good advice about when to be super specific and when not to be specific. My advice is honestly, you do your best, right? If you think that it's in the cristae, go ahead and tell them the cristae. That's what you think, right? If you are on the fence, you're not really sure, you can step back a second and hope that they'll just give it to you for mitochondria. But I usually am one of those, you just jump off the cliff with 2 feet, just get in the water. It's fine. You'll be fine. So just do your best. Again, don't leave anything blank. So you could have said, of course, mitochondria, or you could have said mitochondrial matrix. So part B says to provide a claim how the PDC deficiency affects the amount of NADH produced by glycolysis and by the Krebs cycle in a cell. Provide the reasoning to support your claim based on the position of this in the sequence of the cell respiration, right? So we already know that this works in the Krebs cycle, okay? So let's think for a second, right? NADH, right? NADH is our electron carrier, right? It's going to take those electrons from the glycolysis and the Krebs cycle and bring it to the electron transport chain, right? When it goes from NAD plus to NADH, it means it's holding that electron, okay? PDC works in the Krebs cycle. So in terms of the order of cellular respiration, where is glycolysis? Is glycolysis before or after the Krebs cycle? Glycolysis is before. So if there's a problem with something that's going on in the Krebs cycle, it's not going to affect glycolysis, right? If there's a problem in this thing that converts us from pyruvate into acetyl CoA, that doesn't have any effect on glycolysis. Glycolysis was a step that occurred before, right? So this has ensured that you understood which steps take place when, right? So the Krebs cycle, so glycolysis, you would find no difference or no change in the amount of NADH. And the reason why is glycolysis is going to continue. PDC is not needed. Or you could say glycolysis occurred before we made pyruvate. Your nemesis Krebs cycle. That's okay. You'll figure it out. So the next thing we have is, again, we're talking about the Krebs cycle, right? Well, PDC affects the Krebs cycle. I can't go from pyruvate to acetyl CoA because of the fact that I have this deficiency. So I'm going to see a decrease in my NADH. Why? Because of the fact that you're not going through the Krebs cycle. If you're not able to convert the pyruvate to acetyl CoA, you don't have that starting material, right? It's important to know what goes in and what comes out. What goes into the Krebs cycle is acetyl CoA. Acetyl CoA is the essential, right? It is what provides the rest of the glucose after you've started to break it down so that you'd be able to complete that breakdown, get all of the energy out of it, and release all those carbon dioxide, release those electrons, get all the stuff out of it, right? If you can't make acetyl CoA, you can't get into the Krebs cycle, and thus there's no NADH made in the Krebs cycle. Duh, you got this. But again, this is asking you to make a claim, right? You've got no clue. Let's assume that you have no idea. There's 3 chances, right? Either there's no effect, it increased it, or it decreased it. Just write something. It's okay if you're wrong, right? Just give us your best shot, right? You can't get any points for a blank answer, but you could potentially accidentally get a point by just guessing decrease or guessing no effect. Even if you don't have that reasoning, at least try. You got this. I believe in you. Remember, you are a penguin and you're dressed for success, right? So our third part is where our genetics comes into play, right? So PDC deficiency is caused by mutations in a certain gene, I don't care, and is located on the X chromosome. A male that has this deficiency and a homozygous female with no history of PDC have a male offspring. Calculate the probability that the male offspring will have the PDC deficiency, right? So first thing we need to think about is sex-linked traits, right? If something is sex-linked, it's located on either the X chromosome or the Y chromosome. This question says that it's on the X chromosome, right? So the female has 2 X chromosomes and a male has 1 X chromosome, right? And this is all male and female assigned at birth, right? We know there's a lot of different ways that we determine biological sex and biological gender, right? So this is all just based on X's and Y's and the let's say textbook way that they do it, with females being XX and males being XY. So when we do this cross, right, we're going to see that the male had this X, right, 1 X, and the female's got 2 X's, right? So here, right here would be the male offspring. That male offspring did not get any X's from the father, right? All the X's came from the mother, right? Here's mama and here's mama, right? So if the male has a problem with his X chromosome, he does not pass it on to his sons, right? Or his sons assigned at birth, right? So there's actually a 0% chance, probably 0. Again, let's pretend you have no idea, right? Just guess. 0, 25, 50, 75, 1. It doesn't matter. Just guess one of them. It's okay if you're wrong, especially if you have no clue. Just guess. If you're wrong, you didn't know in the first place. If you're right, yes, I just got a point. So that is the end of today's. Okay, as a reminder, we will meet next Friday for another FRQ Friday. Who knows what time we'll do it, because Miss Jones always changes the time. So we'll do 2017 number 6, 2016 number 4, and 2013 number 5. And what does that make? That may have been the ones we just did. No, that's not. I don't know. I think that's one we're doing next time. Who really knows? But remember, you're AP Bio Penguins and you're dressed for success. Have a good day. Bye, y'all.